So in this lecture, we are going to study about chemical composition of a virus. As we know the structure of virus, previously we discussed about the structure from the nucleic acid to capsid then envelope. So suppose this is the nucleic acid having few segment, for example, 8 segment in influenza viruses. Then this one is capsid. Then this is the envelope, and this envelope is having some spikes, for example HA, NA. Similarly, if we look at the structure of adenovirus, then these are the knobs, and this is the capsid, and this is the DNA. So chemical composition: number 1, nucleic acid; number 2, protein; number 3, enzymes; number 4, lipid; and CHO, carbohydrates. So these viruses, they contain some enzymes, for example RDRP. This RDRP is RNA dependent RNA polymerase enzyme, which is mainly found in some negative sense RNA viruses. And enzymes, for example, this is the enzymes known as reverse transcriptase. So reverse transcriptase is found in Hepadnavirus and retroviridin. So proteins, nucleic acid, that is the DNA or RNA. So one by one we'll discuss about the chemical composition of a virus. Nucleic acid. So number 1 is the nucleic acid, which is also known as the genome of the virus. So nucleic acid, as we know, it could be DNA or RNA, but never both. Never both. All viral genome are haploid. They are haploid. Haploid means they contain one copy, except there is one which contain two copy. And what is that exception is? That is your retrovirus. Retroviruses, they are having diploid genome. So first point is the all viral genomes are haploid. They contain one copy, except retrovirus having two copy or diploid genome. This viral genome could be single-stranded, double-stranded, or partial double-stranded and partial single-stranded. And this is present in Hepadnavirus, that is hepatitis B virus or duck hepatitis virus. So haploid and diploid genome, they are single-stranded, double-stranded, partial, double or single-stranded. Next point is that genome could be single molecule. It could be single molecule. We call it monopartite. Monopartite. If it is single molecule, for example, this is the double-stranded DNA and it is single. So this is known as monopartite. This genome could be having multiple molecules. Multiple multiple molecules or multiple segments. That is known as multipartite, multiple segments. So segmented genome is mostly found in RNA viruses. For example, this is the influenza virus having eight segment. So this segmented virus, this segment number, it lies between 2 and 12. So we'll start with two segment. We'll start with two segments. Two segments are found in A and B. A and B virus. A means arenavirus and B is bi-RNA or virus. That is your infectious bursal disease or Gumboro disease. Then three segments is your Bonia viridae. Then two segment, three segment, then 6, 7 and 8 segments. 6, 7 and 8 segments. In case of influenza virus A type B, there are 8 segments. In type C, there are 7 segments. There are 7 segments. So influenza type A and B, they have 8 segments, and type C is having 7 segments. After 6, 7, 8, 10, 11 and 12 segments. These segments are found in the family Rhioviridae. In this family, there are some genera where 10 segment is found in orthorhiovirus and orbivirus, that is blue tongue virus. 11 segment are found in rotavirus and some aquariovirus. And 12 segments are found in the genus Coltivirus, COLTI, and Sidorna virus, SEADORNA virus, Sidorna virus. So these are the segmented genome, which start from 2 and up to 12, from 2 to 12. Two segments are found in Arenaviridae and Birnaviridae. 3 segments in Bunyaviridae. 6, 7, 8. Which family? Orthomyxoviridae. A and B influenza, 8 segment. C, 7 segment. 10, 11, 12 segments. They are found in which family? Rhioviridae. And these are the genera under this family. 10 segment in Orthorio and Orbivirus, 11 in Rotavirus, 12 in Colti and Sidorna virus. So their genome is segmented. It is present in following segments. And segmented genome is mostly found in RNA viruses. So genomic segments. We discuss about the segments. These genome segments, they code for proteins. So these segment could be monocistronic or poly or multicistronic. That also known as bicistronic if it code for 2 protein. So monocistronic and bicistronic. Monocistronic means code for 1 and bicistronic means they code for 2 protein. So we'll take influenza 8 segment virus as an example. In this example, segment 1 to 6 segment. 1 to 6, they are monocistronic. monocystronic. And segment 7 and 8, these are 7 and 8 are bicistronic, means they code for two protein. For example, segment 7, it code for two protein, matrix protein 1 and 2, M1, M2, M1, M2. Similarly, segment 8, this segment 8, it codes for NS1 and NS2, non-structural protein 1 and 2. So genome segment may be monocystronic or bicistronic. In case of influenza A virus, 1 to 6, they are monocystronic. For example, segment 1, 2, and 3, they code for single protein. This code for polybasic 2, polymerase basic 1, polymerase acidic. So they code for one protein. Then segment number 4 and 6. 4 code for heme agglutinin and 6 code for neuraminidase. So this HA, this is coded by this one, segment number 4. And NA is coded by segment number 6. So these are the monocystronic segment, means 1, 2, 3. They code for single protein, PB2, PB1 and PB3. 4 and 6, 4 code for heme agglutinin and 6 code for N. These three they together combined and form RDRP, RNA dependent RNA polymerase. So this enzyme is found here in the influenza A viruses, and this enzyme is formed by 3 subunit. This first I is the PB2, second I is PB1, and this mouth is PA. So this monocystronic segment, they form 3 protein. These protein assemble and they form the enzyme RDRP. And HANA, these are the peplomers. So we are discussing about the genome, that is DNA or RNA, but never both. This DNA may be in the form of circular and linear, but RNA, they are usually linear structures. Then these RNAs may be plus or negative, plus sense, minus sense. Plus sense means they are infectious. And minus mean they are not infectious. The reason why they are infectious is because they act as messenger RNA. They can code for proteins. That proteins may be structural protein, NSP, non-structural proteins. But these negative sense RNA, they are not infectious because they do not act as messenger RNA. To become as a messenger RNA, they require or they need something that is known as RDRP, means RNA dependent RNA polymerase. It means RNA is a template and it will form RNA, and enzyme uses RDRP. So this enzyme uses RNA as a template This is the negative sense and this is the positive sense. Now this positive RNA is able to make or synthesize structural protein as well as non-structural protein. So this is the difference between positive and negative sense. It means negative sense viruses, they have RDRB with them, and this RDRB is absent in the host cell. So host cell lack this RDRB enzyme, and this is also absent in the positive cells. So this enzyme is only present in the negative sense RNA viruses. And once they become mRNA, they are able to synthesize structural protein and non-structural proteins. So first, genome or nucleic acid is over. Second chemical which is involved is the proteins. As we discussed, the messenger RNA is able to synthesize protein, and these proteins may be structural protein or non-structural protein. What are the example of the structural protein? Some viruses have one structural protein, some have 2, 3, 4, 5, 6, 7, 8. Some have more than 8. So number one is sarcoviruses. Sarcovirus. They have single structural protein. Then parvovirus, they code for 2 structural protein. Then IBD, infectious bursal disease of poultry, 3 protein, that is VP1, VP2 and VP3. And 4, your example is FMD, VP1, 2, 3, 4, 5, 6, 7, 8. 5 is rabies, 5 is baby. And what are the proteins? G, L, M, N, P. Number 6 is canine distemper. F, H, and rest is L, M, N, P. And number 7 is there is blue tongue. And 8 is your influenza virus. So in case of influenza virus, there are total 10 proteins are encoded, 8 are structural protein and 2 are NSP. 2 are non-structural proteins. So these are some of the example of structural proteins and non-structural proteins, for example in influenza viruses. The role of these NSPN and SPE will discuss when we'll study foot and mouth disease. There is known as DIVA strategy. Differentiating the infected versus vaccinated animal. So in that strategy, we'll discuss the role of NSP and these SP protein antibodies. So after protein, what comes is the enzymes. So influenza viruses, they have 8 segments. Number 7 code for 2 protein M1 and M2. These are known as matrix protein. M1 is located just beneath the envelope and it provides the structural support to the virus. So M1 protein is just beneath envelope. These are the proteins. So M2 protein is nothing but ion channel. It is ion channel. And the location of the M2 is within the envelope structure. So we have M2 blocker. Drugs. antiviral drugs. They block this, and that is known as amantadine. Amantadine. So this virus is having M2 channel, and that is involved in the replication. So if we block this ion channel, we can block the replication of the influenza virus with the drug amantadine. Similarly, we have heme agglutinin and this neuraminidase. These two are also known as structural protein. And what is the role of HA? It is for the entry into host cell. Host cell. So for this virus to enter into the host cell, it requires heme agglutinin, and Na is required for the exit. It is required for the exit. So for this Na, we have Na blocker drugs that is known as oseltamivir, and trade name is the Tamiflu. Tamiflu. So these are the structural proteins. Segment number 8 encode for non-structural protein. Rest all 7 segments they code for 8 structural protein. Out of these 8 structural protein, these are some of the important structural protein. 2 are in the envelope HANA. HA used for entry into host cell, Na for exit from the host cell. If we block this Na with the drug oseltamivir Tamiflu, the virus is unable to come out of the infected cell. So we can prevent the spread from infected cell to healthy cell by blocking this Na. Second is M2 channel. These are two protein M1, M2. M2 is ion channel. We can block this ion with some medicine so that replication will stop. So these were some of the structural protein. Now we'll discuss the enzymes. Which enzyme is present in the influenza virus? Okay. Example of enzyme is RT-RP. Where it is present? It is present in negative sense RNA viruses. So number 1 is negative sense RNA virus. It is present. And then where it is absent? Host cell. So host cell lack this RD-RP. It is absent. What is the composition? Which of the segment encode for this RD-RP? This RD-RP is a heterotrimer made up of 3 subunit. It is made up of 3 subunit. This I is the PB2, this is PB1, and this is PA. These 3 subunit make the enzyme RD-RP. And these 3, which segment code for this? This is coded by segment 1, this is by segment 2, and this is by segment 3. So this is the structure of RD-RP of which virus? Influenza A virus. Similarly, rabies is also negative sense. Canine distemper is also negative sense. Both have RD-RP, and that is made up of 2 subunit, L subunit and C subunit, L and C. So L protein is the core and P protein, P is the cofactor. So this here the RD-RP is made up of 2 parts. The core is the L protein. Core is the L protein, and this cofactor is the P protein. P protein. So this is how rabies and canine distemper RDRP is composed of L protein and P protein. And the role of this RDRP is to convert negative sense into positive sense and make it more infectious in nature. So negative RNA, how to remember this? You can remember very well from the two example or two vaccine that we are using in our day-to-day life. One is DHPPI. It is having two DNA virus and it is having two RNA viruses. Second vaccine we are using is the rabies. So these are the two most commonly used vaccines in the small animals. DHPPI vaccine and rabies vaccine. So the two RNA viruses in the DHPPI, they are negative sense. They are negative sense, and the rabies virus itself is the negative sense. So these are the most common way to remember these three RNA negative sense RNA viruses. So last portion is the lipid and carbohydrate. So where the lipid is present, this is the envelope of the virus, and it is lipoprotein in nature. Lipoprotein nature. This lipid portion is derived from cell membrane of a host, and this carbohydrate portion is also derived from the cell membrane of the host. Then the subunit of this envelope, HA and NA, that is known as peplomers. And chemically their composition is glycoprotein. They are glycoproteins. So now we'll discuss the role of these two peplomers which are a part of the envelope. So there are two phenomena, heme agglutination and HAD, heme adsorption. So this phenomena, it requires red blood cells and virus. There is role of RBC and virus. In this phenomena, there is role of red blood cells, then virus and virus infected cells. For example, macrophage. In case of the virus could be African swine fever, and the infected cell is the macrophage. So heme agglutination. Heme agglutination means heme means RBC and agglutination means their clumping. So this RBC clumping, it could be direct. For example, this is the adenovirus having here is the knob and it interact with the red blood cell receptors. Because of this interaction, there is direct agglutination of the RBC. So example is adenovirus as well as parvovirus. And number two is via heme agglutinin. Heme agglutinin means this one HA. So there are some viruses which use heme agglutinin to agglutinate the RBC. This is the RBC having receptor sialic acid on the surface. So this HA react bind with this one and clumping of RBC. So there are two examples via this heme agglutinin protein, which is nothing but a glycoprotein, glycoprotein or spike or papillomer. Number 2 is Newcastle disease, that is Ranikhet disease. So these two viruses, they use their heme agglutinin glycoprotein and cause heme agglutination, clumping of the RBC. Adenovirus, parvovirus, they directly agglutinate the RBC. They do not have any HANA glycoprotein on their surface. Now, concept of heme adsorption. AD means adsorption. Adsorption means a phenomena which happens on the surface. Here, what is the surface? Here, surface of some infected cell, and these viruses are enveloped viruses. These are the enveloped viruses. I can say some enveloped viruses, they enter the infected cell and they incorporate some protein in their cell membrane. These are the incorporation. For example, in case of African swine fever, this is CDV protein, and this is the protein which cause this RBC to stick on the surface, of the infected cell macrophage. So this is the cell. Over the surface, the RBC are clumped together. It look like a rosette around the infected cell. So this is known as heme adsorption. Adsorption means on the surface of which cell? Infected cell. For example, macrophage in case of African swine fever. And these enveloped virus, they implant, they insert, they incorporate some glycoproteins into the cell membrane of the host cell, infected cell. And these proteins, then they clump the RBC, they bind the RBC on the surface of the cell. So we require three things here: RBC, infected cell, and the virus. And which families show this kind of heme adsorption property? Just remember, for example, orthomyxo, paramyxo, toga virus, some toga virus, and your African swine fever. So this is the difference between heme agglutination and heme adsorption. This could be direct, like adeno and parvo. Indirect means use of some heme agglutinin, influenza, Newcastle disease. But here what is required is the surface, because it is a surface phenomena, and the surface is of infected cell, and infected cell is having some proteins, and these are the viral glycoprotein which are inserted into the cell membrane of the macrophage. Now these interact with the RBC. So they interact with the RBC. So multiple RBC will stick over the surface. They look like a rosette, like petals of a rose flower. So these are the two properties of envelope spikes, papillomers, or we can say envelope glycoproteins: heme absorption, heme adsorption, heme agglutination, heme adsorption. In future lecture, we'll discuss about heme agglutination inhibition assay, HAIS. Similarly, heme adsorption inhibition assay.